Hi, and welcome to another episode of Tom Kennedy Science. I'm your host, Dr. Tom Kennedy. Now today, what we're going to talk about is apoptosis. Now some people, they do pronounce that apoptosis. I think that's actually the correct way, but it's the way we say it in society is apoptosis, which is programmed cell death. Whoa, programmed cell death? I find it amazing that basically every animal on this planet has a death switch programmed into them. And if they receive the correct signal, they will commit suicide. And they can also have problems with them internally that can also send them down the path of apoptosis, which is pretty amazing. Oh, there I go, apoptosis, apoptosis. Now, apoptosis is not that common in a lot of introductory textbooks, but it should be. This is one of the greatest inventions in evolution. Wait, hold on. How in the world is death a great evolutionary innovation? Well, actually, it's very important in the evolution of multicellular life. Okay, so we need it for growth and development. And I'll talk about that. We need it as a way to prevent cancer. We need it as a way to maintain homeostasis. Did you know that every day you lose between 50 and 70 billion cells? That's a lot. And you lose them through apoptosis. Now, of course, apoptosis is heavily regulated because once a cell commits down the path of apoptosis, it's committed. Like once it's triggered, there's no going back. It's like setting off the self-destruct in a spaceship, right? I mean, except in Star Trek, it's kind of they always manage to stop it at the last second. Not in apoptosis. Once you start it, you're committed. And this is important because if you have excessive apoptosis, that leads to atrophy. That leads to muscle wasting. And if you have a lack of apoptosis, that leads to excessive cell growth. Well, that sounds pretty bad, doesn't it? Because excessive cell growth is cancer. And a lot of people consider cancer as basically a failure of apoptosis. So you can imagine that this is really, 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 really important to study. And in biology, we have model organisms, mice, zebrafish, yeast, E. coli, the bacteria in your gut, and worms. And in fact, a lot of what we know about apoptosis and the apoptotic pathways are from this C. elegans. Cenorhabditis elegans. It's a tiny little microscopic roundworm. Yes, I know we study these things and we learn so much about ourselves by studying these things. And the reason why we can do that is because of the shared common ancestry of all animals. See, all animals had a single common ancestor. And paths like apoptosis are conserved more or less across the animal kingdom because we inherited them from our common ancestor. Kind of cool, isn't it? Okay, so apoptosis, this programmed cell death. Now, this can come from a death signal outside the cell. We would call that an extrinsic pathway. So you can imagine you've got these macrophages. Macro means big, phage means eater. These are white blood cells or a type of white blood cells that are patrolling your body, and they can release a chemical like a cytokine. Now, for those of you into the coronavirus, you know that part of the problems that makes people have very bad reactions to the virus is a cytokine cascade. But cytokines are a protein signaling molecule. And what these macrophages will do is they can release a signal to a cell that's like not doing well or it's time to go, and that can be a death signal. And since it's coming from outside the cell, that is an extrinsic pathway. And that's what we're going to focus on today as an overview. And the other way, of course, is the intrinsic pathway, and that is also known as the mitochondrial pathway. And that has to do whenever the cell becomes stressed, might be infected with a virus, or it may have real problems with its DNA. It can internally set off apoptosis through the intrinsic pathway. And if you notice, the mitochondria involved with this are endosymbiont. So, another reason why I think mitochondria is so cool, right? Because they're part of the evolution and the process of programmed cell death. And in fact, they'll release cytochrome C into the cytosol, and that can initiate apoptosis. And if you remember, cytochrome C, it's an electron carrier, and it carries electrons between complex 3 and complex 4. Okay, now, Whatever this death signal, whether it's an extrinsic death signal or the mitochondria is triggering it, and the mitochondria trigger it when they lose membrane potential. So when mitochondria aren't doing well, they can set off this signal here. And what they do is they activate these caspases, right? And caspases are a class of proteins that have multiple purposes within the cell, which we'll come to in a second. It also involves another class of proteins called the SED or CED proteins as well. And whether we turn those on or off, and that, like I said, leads to this signaling cascade within the cell committing it. But let's talk about caspases for a second, because they're kind of interesting. Now, whether you're intrinsic, you're activating apoptosis from within, or it's an extrinsic, you're getting the death signal from another cell, like a macrophage, you ultimately turn on caspases. Now, caspases come in two different broad categories. One are the initiators, and these are, once again, signaling molecules. And they're important because once you've activated them, they have important roles in inflammation, and they have important roles of bringing immune cells to areas that are infected, hence inflammation. And that's why you get pussy, because you're bringing in all these, cells to help clean that up. And so these are important. And then you also have, I love the name of this, the executors. You can imagine what executors do. Executors include proteases. Protease, ACE, there it is again. There's an enzyme. Proteo, protein. Yes, these are enzymes that break down proteins. And there's multiple different types inside of your cells, and they can break down like over 600 different cellular components as the cell is preparing for death, which this programmed cell death, which is very orderly, and that's important. Now, one other thing, these caspases, these executors, they're important, for breaking apart all these different proteins inside the cell. And the initiators will also turn on nucleases, specifically DNA-ase, which will then chop up all your DNA. This is important, right? So if you think about it, cancer cells are a proliferation of our cells. They're damaged. They're not going through our normal checkpoints. They're growing too much. And so you can imagine that a caspase deficiency has been involved with the proliferation of different types of cancers. So let's talk a little bit more about the extrinsic pathway here. You get the death signal. And I mean, these things all have specific scientific names. I'm not going to go into here what exactly their names are, but it's a ligand, right? It's a chemical signal, and it comes in and it binds to a receptor on the membrane, okay? And then that receptor will then transmit the signal into the cell. Now, normally there's a protein, it's called SED9, and it's inactive. And what it's doing is it's suppressing the entire apoptotic pathway. And once SED9 becomes activated, it goes and activates two other proteins, SED3 and SED4. Oh, are we seeing like an amplification and a cascade going on here? You bet. And what those go on to do, guess what? They activate the caspases. So all routes lead to the caspases, both the initiators and the executors. And those initiators can now start doing their thing of cell signaling, and they initiate the sum of the cell signaling to coordinate the efforts around that cell and inside the cell. And they also turn on the executors, which then start to chop up all the proteins within the cell, including taking apart the cytoskeleton as well. And then the nucleases, which are also activated by other types of caspases, they begin chopping up the DNA. Now, that makes sense, because if you've got a viral infection, one way you can stop a viral infection, well, viruses, what they do is they take their DNA or RNA, they can be either one, depending on the virus, they insert it into your cell, and they take over the machinery of your cell, right? So they basically use their DNA or RNA to start making more viruses. So if you're releasing nucleases, or if you're activating nucleases, you start chopping up that DNA, including your chromosomal DNA, and you can prevent additional viruses from spreading. Now that's kind of the basic view of this. I just want to show you this chart. Okay, I know I always look at stuff like this, whether it's a pathways for apoptosis or it's a pathway for all of our cell metabolism. And I look at these and I'm just in awe. The amount of work and knowledge that has gone into creating something like this. And it's not a function of just trying to memorize it. It's a function of like, this is what people have figured out. And to me, that's important to realize, that when we teach something in an intro biology course, we're getting the surface of it, right? We're getting the very basics of something, and we're going, wow, that's kind of complicated. The reality is, it's actually way more complicated than the way we talk about it in these intro classes. And I like to remind people that, man, there's still a lot going on, and there's still a lot that we don't know. Okay, so here it is, apoptotic pathways, intrinsic or extrinsic. They activate these caspases, and they can be triggered by an extracellular event, a signaling ligand, the death signal, damage to the DNA nucleus, and also another one is as our cells begin to age, one of the stressors we get is we build up oxidative damage. And also, as our cells age, they don't fold proteins correctly. And that often happens in the endoplasmic reticulum because any protein is going to be exported outside of a cell, embedded in a membrane, or inside of another organelle. Well, they go through the endomembrane system, especially the endoplasmic reticulum, where you've got all these chaperonins and other proteins to help these proteins fold. And once they start not working so well, guess what? It sends a signal and the cell can kill itself. And that's part of maintaining our homeostasis, right? I told you we get rid of 50 billion cells a day, some of us even 70 billion. We do that so that we can maintain, new cells that are functioning correctly. And one other reason why this is important, we talk about apoptosis for preventing, disease, spread, cancer, but it's also important for growth and development. I know, you don't think of babies as growing or even an embryo growing and needing to kill cells off, but they do. And in fact, early on in our development, we all start off with our hands and they're webbed, and you can see that interdigital tissue. And I can't imagine why we would ever start off with webbed hands. Something about we evolved from fish? No, seriously, we evolved from fish. We are a modified fish. Our fish ancestor was a lobed fin fish that lived about 375 million years ago. So if these are a modified fin, it makes a lot of sense to me that our digits would have webbing between them. Well, early on in our development, apoptosis comes along and kills the cells between our digits, and we actually have digits without webbing between them. That's pretty cool, isn't it? Those are not the only cells we lose in our growth and development as our brains grow, right? As a baby, and I'm watching my little 3-year-old, her brain is growing by leaps and bounds. Not only is it actually adding more neurons, and then once they hit adolescence, guess what? They start losing brain cells rapidly. No, that's not why teenagers are dumb. No, teenagers are actually quite smart. And in fact, they can do things that us adults don't do as well. But the reason why a teenager starts losing brain cells is because the brain creates way more than it needs. And then as you start using your brain, learning and whatever, it will use the neurons that it needs. And then the ones that aren't being used are basically die through apoptosis. And that actually streamlines it and makes the brain work faster. So it's actually a good thing. We never think of losing brain cells as a good thing, but in actuality, in our development as we become an adult, it is a good thing because it helps streamline our brain, make us think faster. And also, growth and development. We've talked about disease as well. Some other diseases, not just cancer, but Parkinson's, Alzheimer's, there may be interference with apoptotic routes that are involved with that. So, like I said, this is still an active area of research. Apoptosis, even though we've known about it for almost 30 years, you're looking for a job, this is a good place to go into. And then I've already talked about this, but I've got to bring it up again. The evolutionary significance of apoptosis. It allowed for the evolution of multicellular life. You can see it now. I've talked about it. Growth and development. We can tell cells when to die when they need to. We can have cells die throughout our life to make way for newer cells that are working more correctly. And we can also use apoptosis to protect us from cancer. Cancer is the price we pay for being a multicellular organism. We have 17 to 20 trillion cells. You have to control when they grow, when they divide, when they die. And if we can give us a death signal to a cell, we can prevent cancers from spreading. And then when apoptosis goes wrong, we can get cancer. If it's overdoing it, we get atrophy. Other problems might involve Alzheimer's disease, Parkinson's disease. So like I said, this is, even though it's not in our textbooks very much, apoptosis is incredibly important and actually a really cool thing to study. Okay, well, this has been another episode of Tom Kennedy Science. Until next time.